Good day, everyone. I am Michael Allen M. Mendoza, and our group chose viral transcription of COVID-19 as our topic for the final output in molecular biology and diagnostics laboratory. This presentation will tackle the methodology of our chosen research paper relating to viral transcription of COVID-19. But first, we will provide a brief background or introduction about COVID-19.
Good day, everyone. I am Vilmarama, and today I'm going to give you a brief background about COVID 19. So, first, what is COVID 19? It is a severe acute respiratory syndrome, or also known as coronavirus SARS CoV 2, a new virus recently named by the Coronavirus Study Group of the International Committee on Taxonomy of Viruses. And it originated in Huanan Seafood Wholesale Market in Wuhan, China. So, it's the epicenter of the infection. Wuhan, China. So its symptoms include fever, myalgia or muscle pain, cough, dyspnea or difficulty in breathing, radiological evidence of ground glass opacities compatible with a typical pneumonia. So as you can see in the X-ray result of the COVID 19 infected people or patients, you can see a hazy or glassy like result in the x-ray. So for the reported cases, it is a person-to-person transmission or infection, mostly about close family contacts and healthcare workers. These are the people that are very prone to infection because of the close contact every day. And as far as I can remember, the first case in the Philippines was reported on January 30 this year, 2020. And the epicenter of the infection is in San Juan City, Green Hills. So the next one is it is highly sensitive and specific laboratory diagnostics for COVID-19, and they are essential for case identification, contact tracing, animal source finding, and rationalization of infection control measures. And the following are the limitations for the diagnosis of COVID-19. So, since this is a new virus, there are a lot of limitations for its diagnosis because the experts are still finding ways on how to detect this novel coronavirus. So first one is the use of viral culture for establishing acute diagnosis, which is not practical. And then the next one is isolation of the virus that requires biosafety level 3 facilities, which are not available in most healthcare institutions. So as you can see here, it's about the healthcare problems and the biosafety level cabinet that will be used in detecting COVID-19. And lastly, serum antibody and antigen detection tests have not yet been validated, and there may be cross-reactivity with SARS-CoV. So there are a lot of variety of SARS-CoV. So right now we have this novel coronavirus or SARS-CoV-2, and they are like a family. So the results may be mistaken as coronavirus kahit hindi naman. So the gold standard for laboratory diagnostic tests for COVID-19 is reverse transcription polymerase chain reaction or RT-PCR. And on January 23, 2020, the protocol of the first real-time PCR assays targeting the RNA-dependent polymerase, envelope, and nucleopatid NI genes for SARS-CoV-2 were published. And then the assays, probe 1, pan-Cerbeca probe, which would detect SARS-CoV-2 or COVID and bat SARS-related coronaviruses, and probe number 2, RoRo-P2, specific for SARS-CoV-2 and should not detect SARS-CoV. These assays are designed and validated using synthetic nucleic acid technology and in the absence of available COVID-19 isolates of original patient specimens. So these are the tools or the assays that we use to detect specifically the novel coronavirus. Thank you.
Objective: the study aims to develop and compare the performance of three novel real-time reverse transcription PCR assays targeting the RNA-dependent RNA polymerase, helicase, spike, and nucleocapsid genes of SARS-CoV-2.
The World Health Organization was alarmed by an abrupt increase of cases of pneumonia in Wuhan, and was soon found out that the cause was COVID 19, also known as Severe Acute Respiratory Syndrome Coronavirus 2 or SARS-CoV-2. It is a part of a family of coronaviruses, which are a strain of single-stranded RNA viruses. Patients infected with the virus manifested symptoms such as fever, cough, myalgia, dyspnea, and pneumonia. Some patients do not exhibit any of the aforementioned symptoms and are labeled as asymptomatic, or have mild symptoms and are called mildly symptomatic. The detection or diagnosis for COVID 19 must be highly sensitive and specific in order to handle the pandemic properly, as seen during the previous pandemics such as SARS-CoV and MERS-CoV. In the present, reverse transcription PCR or RT-PCR remains the method of choice as other techniques like viral culturing and the likes have issues with practicality, availability, or validation. The protocol for RT-PCR was released in January and targeted the RNA-dependent RNA polymerase, the envelope, nucleocapsid genes of the virus, with RDRP having the most analytical sensitivity until the development of a better real-time RT-PCR, which will be explained later.
Good day, everyone. I am Vilmarama, and today I'm going to discuss with you the model for coronavirus replication and transcription. So, to make it easy, mechanistically speaking, the process of discontinuous transcription during minus strand synthesis can be viewed as number of consecutive events. So, as consecutive events, we have the first one, which includes that the components of a functional RTC, or the RTC stands for replication and transcription complexes. This is the term that we're going to use to describe complexes copying or producing genome. So copying or producing genome. So it's in the complex itself, or the term RTC, or subgenome length RNA. These complexes accumulate at perinuclear region and are associated with double membrane vesicle according to the journal. So going back, so first one is the components of functional RTC are recruited and minus strand synthesis is initiated at the 3' end of a genomic RNA. So the genomic RNA in here in the picture is the red line that you can see. So genomic RNA. So as you can see, the RTC, which was represented by the teal color or the oval, yung mga bilog-bilog na yan, were then recruited at the 3' on the right side. Kita nyo? Diba? Lahat sila na-move dun sa right side. As you can see. So, that's the first one. And then the second one is the elongation. So, be mindful about this. The elongation of nascent minus strand RNA continues until the first functional body, TRS. The TRS is what we call transcription regulatory sequences. Motif is encountered. So, TRS would either stop or continue the synthesis of the nascent minus strand. So, a fixed proportion of RTCs will either, ito nga yung sinasabi ko, will either disregard the TRS motif and continue to elongate the nascent strand, or pag naman nakilala nung group of RTC si TRS, so ano ulit si RTC? Replication Transcription Complexes. So, ito yung nagsasabi ng copying of the genome and producing the complexes. So, yeah. So, kapag na-encounter niya si TRS, ano nga ulit si TRS? Transcription Regulating Sequence. So, kapag nakilala niya to, the synthesis will stop at the nascent minus strand and relocate and complete its synthesis somewhere else. So, ito nga yung tinatawag natin, as you can see here in the picture, it's discontinuous, diba? Hindi tuloy-tuloy. Kasi nga, it's either maging continuous siya or tuloy-tuloy siya mag-synthesis kapag dinisregard niya si TRS, or maging discontinuous siya kapag nakilala niya si TRS, and then it would relocate somewhere else. Kaya, as you can see here, tingnan mo, hindi pantay-pantay yung mga RNA nascent minus strand natin, which was represented by the blue line in the picture. So, and then the last one, this relocation will be guided by complementarity between the 3' end of the nascent minus strand and the leader TRS motif. So, magsishift siya doon sa from parang polar to polar, opposite to opposite. So complementary nga, 'di ba? 3' to 5', 'di ba? The translocated manostrand will be extended by copying the 5' end of a genome. So yung 5' end ng genome or nung red line na nakikita niyo dyan sa picture, yun naman yung kakopy niya. So saan nga ulit to yung copying na step? It can be seen in the step 2. So elongation of nascent manostrand. So nag-elongate ulit siya by copying the 5' end of the genome. The completed manostrand RNA would then serve as template for mRNA synthesis. So all of them would be the complete form would be the manostrand RNA that would serve as a template for mRNA synthesis. In continuation, as a reinforcement to my discussion a while ago about the model for coronavirus replication and transcription, let us analyze thoroughly the model. So I want you to look first at the topmost here. So the ORF1. So what is an ORF1? ORF1 is the opening reading frame. So this would signal the start of the whole process. So the ORF1 of genomic RNA, which was designated the red line as you can see here in the model, RNA red, is translated to produce. So this red line is translated to produce PP1A and PP1AB. So what are these two? These two are large polyproteins, and then the synthesis of PP1AB involves programmed ribosomal frame shifting during translation of ORF1A. During or after synthesis, these polyproteins are cleaved by the virus itself. Virus-encoded proteinases with papain-like PL and chymotrypsin-like folds into 16 proteins. So these two polyproteins are essential for the viral proteins. Yeah. And then so after they were translated to PP1 to produce PP1AB and PP1AP, it would assemble into an RTC. What is an RTC? Again, RTC is the term that we uses to describe complexes copying or producing genome. So they either copy or produce genome. So RTC stands for replication transcription complexes. So yeah, it was in the form of the oval shape or the teal oval that recognizes cis acting elements at the 5' and 3' ends of the genome. So this one are those recognize blah blah the 5' and 3' ends, and then this RTC copies the genome either continuously into genome length template or discontinuously into the various subgenome length minus strand template. So how does the process become continuous? It became continuous if the RTC or the group of RTCs disregard the TRS motif, like what I said a while ago. What is the TRS motif? Transcription regulating sequence. So if they didn't notice or hindi nila nakilala ang TRS, they would disregard that and continue synthesizing until they become RNA synthesis. So if not, here, as you can see here, so it would become discontinuously into the various subgenome length minus strand templates. The subgenome length minus strand templates are represented by the blue ones, the blue lines under the red one. Yeah. The minus strand blue, like what I said, are used as templates for genomic and subgenomic mRNA synthesis. Only genomes are used as templates for minus strand synthesis. Example is in replication, and then the RTC or the replication transcription complexes engaging in plus strand synthesis age and release their minus strand templates, which are then degraded specifically. So that's the whole process or the model for coronavirus replication and transcription. The final product would be subgenomic mRNA synthesis. That would then repeat the process.
This slide shows the protocol or flowchart of our methodology. It consists of specimen collection, reagent preparation, nucleic acid extraction, in vitro RNA transcription, COVID-19 real-time RT-PCR, RT-PCR sample processing, RNA amplification, RNA analysis, and statistical analysis.
For the specimen collection, SARS-CoV-2 was isolated from a patient with COVID-19 in Hong Kong. Archived laboratory culture isolates were gained from the Department of Microbiology in the University of Hong Kong. The 273 specimens obtained were from respiratory tract and non-respiratory tract. These are all from 15 patients who suffer from COVID-19. It is also important to do this experiment in a biosafety level 3 facility because it may cause lethal disease through inhalation and may contaminate the environment. Additionally, total nucleic acid extracts of 22 archived nasopharyngeal aspirates were also acquired.
For the reagent preparation, we will be using primers. Total nucleic acid will also be used as template because total nucleic acid extraction products purify both RNA and DNA from samples. We will also be using RNase-free water and RT-PCR master mix. For the preparation of master mix, it will be discussed on the preceding slide. On the table on the screen, it shows the primers needed for the procedure. FIP and BIP. It is designed to hybridize the complementary and reverse complementary targets of F2 or B2 and F1 or B1. Next primer is the F3 and B3, which can recognize a total of 6 distinct regions within the target DNA or DNA. And lastly, the forward loop primer and the backward loop primer. It is used to accelerate the amplification and detection and efficiency.
For the master mix preparation, each 20 microliter reaction mixture contains 10 microliter of 2 times Quantinova probe RT-PCR master mix, 0.2 microliter of QN probe RT mix, 1.6 microliter of each 10 micrometer forward and reverse primer, 0.4 microliter of 10 micrometer probe, 1.2 microliter of RNase-free water, and lastly, 5 microliter of total nucleic acid (rTNA) as the template.
Nucleic acid extraction. Total nucleic acid extraction of specimen and cell culture of viral isolates were performed using a Nuclisense EasyMag extraction system. The volume of the specimen used for extraction and the elution volume dependent on the specimen type and the available amount of the specimen. In general, 250 microliter of each respiratory tract specimen, urine sample, fecal swab, and feces were subjected to extraction with an elution volume of 55 microliter, and 100 microliter of each plasma specimens were subjected to extraction with an elution volume of 25 microliter. The extracts were stored at negative 80 degrees Celsius until use. The same extracted product of each specimen was used for all the RT-PCRs. In vitro RNA transcripts for making positive controls and standards. Linearized PCR 2.1 topo plasmid with a T7 promoter and a clone target region of SARS-CoV-2 were used for in vitro RNA transcription using Megascript T7 transcription kit. Each linearized plasmid template was mixed with 2 microliter each of adenosine triphosphate or ATP, guanosine triphosphate or GTP, cytidine triphosphate or CTP, and uridine triphosphate or UTP, 10 times reaction buffer, and lastly, enzyme mix in a standard 20 microliter reaction mixture. The reaction mixture was incubated at 37 degrees Celsius for 16 hours, followed by the addition of 1 microliter of turbodinase, and was further incubated at 37 degrees Celsius for 15 minutes. The synthesized RNA was cleaned by RNA-seq minikit. For quantification, biodrop U-Light was used to quantify the concentration of purified RNA.
The real-time reverse transcription polymerase chain reaction assays for SARS-CoV-2 RNA detection were performed using Continova probe RT-PCR kit in a light cycler 480 real-time PCR system. The sample preparation for real-time reverse transcription polymerase chain reaction includes RT-PCR buffer, water, deoxynucleoside triphosphates, gene-specific powder, reverse transcriptase, DNA polymerase, and RNA template. For the sample processing, the thermal cycling condition was 10 minutes at 45 degrees Celsius for reverse transcription, 5 minutes at 95 degrees Celsius for PCR initial activation, and 45 cycles of 5S at 95 degrees Celsius and 30S at 55 degrees Celsius.
For the RNA amplification, a loop-mediated isothermal amplification assay, or LAMP, that amplifies DNA with high specificity, efficiency, and rapidity under isothermal conditions was used as a rapid diagnostic for COVID-19. According to L. Tolot, there are two-stage lump strategy, which could be carried out in closed tubes with either fluorescence or colorimetric detection, and using a visual colorimetric in identifying COVID-19 viral RNA from purified RNA or patient cell lysis was said to be successful in detecting the presence of COVID-19. For the procedure, a sample must be collected first. It can be either from swab or blood samples. Once a sample is collected, transfer the sample from one tube to another and heat the tube at approximately 60 to 65 degrees Celsius. Once the tube is heated, transfer it to absorbent tube and freezing of dried lump master mix. Subsequently, mix the sample solution with the LAMP master mix powder and dispense it into multiple tubes. And now perform the LAMP reaction. The results via real-time detection, colorimetric and fluorescent dye, and lateral flow dipstick. For demonstrating the outcome of the lump assay using a colorimetric change to detect the presence of COVID-19, the darker color represents a positive assay while the lighter color represents the negative assay result.
Next, RNA analysis. The other discrepancies seen in the results were analyzed and confirmed by testing it with light mixed modular SARS, Wuhan COVID e-gene kit, and bat SARS-like coronaviruses. They are both rapid and sensitive for SARS-CoV-2 detection. They also have fewer verification requirements compared to laboratory-developed tests. The overall analysis was enunciated through the idea of each 20 microliters mixture contained 4 microliters of Roche master, 0.1 microliters of reverse transcriptase enzyme, 0.5 microliters of reagent mix, 10.4 microliters of water, and 5 microliters of TNA as the template. In the statistical analysis part of the journal article, Fisher's exact test was the main tool used by the researchers in order to compute for the population who tested positive for RDRP2. Fisher's exact test is a statistical test used to determine if there are non-random associations between two categorical variables. Moreover, if p-value is less than 0.05, then it is statistically significant. Additionally, it was analyzed through GraphPad from software.
In the detection of SARS-CoV-2 RNA in clinical specimens, the authors evaluated the performance of two assays. So these are the COVID-19 RDRP helicase assay and RDRP-P2 assay. So with the results, a total of 120 respiratory tract. These specimens are collected from nasopharyngeal aspirates, throat swabs, saliva, and sputum specimens, and 153 non-respiratory tract specimens, which are collected from plasma, urine specimens, feces, rectal swabs. So these specimens are collected from 15 patients with laboratory confirmed COVID-19 in Hong Kong. Among the 273 specimens collected from those 15 patients, 77 were positive by RDRP-P2 assay. So as you can see on the table, RDRP helicase assay was also positive for all of those 77 specimens. So both of the assays are positive with those 77 specimens. But additionally, RDRP helicase assay was also positive for another 42 specimens, making it 119 out of 273 specimens are positive for RDRP helicase, while with RDRP-P2 assay, it only has 77 out of 273 specimens that are positive. All of those 42 additional specimens with on RDRP helicase, they are confirmed positive by light mix modular SARS and Wuhan CoV-E gene kit with the light cycler multiplex RNA virus master. So 29 out of 120 respiratory tract specimens and 13 out of 153 non-respiratory tract specimens are negative by the RDRP-P2 assay. The mean viral load of those specimens was 3.21 times 10 raised to 4 RNA copies per milliliter and was about 6-fold higher in the respiratory tract specimens than the non-respiratory tract specimens, since the respiratory tract specimens only have 4.33 times 10 raised to 4 RNA copies per milliliter, while the non-respiratory tract specimens have 7.06 times 10 raised to 3 RNA copies per ml. So, therefore, the COVID-19 RDRP helicase assay was significantly more sensitive than the RDRP-P2 assay for the detection of SARS-CoV-2. In nasopharyngeal aspirates or throat swabs, saliva, and plasma specimens. So as shown in the figure, the COVID-19 RDRP helicase assay consistently detected SARS-CoV-2 RNA in those samples with a higher sensitivity than the
RP2 assay throughout the patient's course of illness up to day 12 to day 18. The sensitivity of the two assays did not differ significantly for sputum specimens and feces or rectal swabs.
Due to these circumstances only happening recently, it is obvious that studies still need to be conducted in order to learn about the coronavirus. Therefore, there are insufficient in-use evaluation data of the stated assays that used a large number of clinical specimens from patients with confirmed COVID-19. In this paper, the researcher developed and evaluated three novel real-time RT-PCR assays that tackled different gene regions of the SARS-CoV-2 genome. Also, the researchers demonstrated that the novel COVID-19 RDRP HEL assay was very specific and sensitive for identifying the SARS-CoV-2 RNA in vitro and in COVID-19 patient specimens. In addition, acquiring coronavirus enzymatic activities help the fidelity of RNA replication and transcription, thus allowing genome expansion. As a result, the coronavirus have the capability to extend their host range and easily adjust to changing environmental conditions while regulating genomic stability. The aim of the study is not necessarily a quantitative measure of infection, but rather a simple positive or negative assay for rapid detection or confirmation. We consider that this strategy should be applied rapidly and confirmed for viability with clinical samples before being rolled out for mass diagnostic testing in these current times.
On the other hand, the loop-mediated isothermal amplification, also known as the LAMP assay, showed that it can be used as a rapid diagnostic test for COVID-19. A LAMP assay is simple and portable, which doesn't require a specific skill to a health worker. Therefore, the operator no longer needs to undergo a long-term training just to be qualified to perform the assay. It is also cost-effective, and the reagents costs are just the same as the real-time RT-PCR. And the biggest advantage of this assay is its time efficiency, because unlike RT-PCR, which takes 4 to 8 hours before the results can be released, the LAMP assay result will only take an hour to be generated.
Comparison between PCR and LAMP-based methods of viral RNA detection by Nguyen et al., as cited by Kashir and Yaginuddin, is shown in this table.